Today we're featuring Craig Venter, a world-renowned biotechnologist known for his groundbreaking contributions to genomics. He had a pivotal role in leading the first draft sequence of the human genome and assembling the pioneering team that achieved the transfection of a cell with a synthetic chromosome.
Cells are very dynamic, changing second to second. But one thing is fundamental to all life, and that's the genetic code.
Later in life, he and his research team created the world's first synthetic organism from scratch, demonstrating the potential of synthetic biology to engineer life at the molecular level.
Taking those gene pathways, putting them in synthetic organisms, we'll be able to create whole new chemical libraries.
Craig's a leading figure. He's hilarious, unfettered, unshackled, and unafraid. You'll hear all of that in this episode and find out why he was among Time Magazine's 100 most influential people in the world. Not once, but twice, maybe on another planet where life already exists. So without further ado, we welcome a lively episode of the Into the Impossible podcast. My friend Craig Venter, live, recorded at UCSD this past fall.
Welcome back to our extended session with none other than Dr. Craig Venter, whose incredible intellect and influence on so many of us. And I wanted to solicit questions, and so I did take them on Twitter, X, and also on Instagram and on my YouTube channel. Dr. Brian Keating is the handle for all those. So we're going to get some wild and interesting stuff, ranging from panspermia to UAPs to life on other planets. But we're going to begin with a more close to home. Okay, so the first question. Hi, Brian. And this is from Dr. Thomas Geraj. It may be interesting to talk about cancer and the somatic mutation theory. Any ideas why there has been so little to no advance in survival for most advanced solid tumors, despite our understanding of the human genome? Do you believe, Craig, that cancer is purely a genetic disease?
Well, it's definitely a genetic disease, but that doesn't mean its ultimate cause is genetics. Radiation causes cancer by mutating the genome. Mm-hmm. So it's the radiation that causes the cancer. Same things happen with chemical exposure, UV light. I'm on my, I think, 12th or 14th round of Mohs surgery. The surgeon said, have you been exposed to sunlight? I said, for 77 years, and I was exposed to sunlight long before there was sunscreen. So I have more than my share of skin cancer from UV light exposure.
My father-in-law had acne treated using X-rays back in the 60s.
Yeah, and X-rays are definitely a carcinogen on their own. So, but these things change the genetic code. So at human longevity, we would sequence the individual's genome and sequence the tumor's genome. And you find all the genes that are mutated. And it turns out these can be very useful, and a lot of therapies are being developed on. The term we use is neoantigens because it's a mutated protein. It actually changes the structure of the protein, and the immune system can recognize it. And there's been attempts to take these out and see which T cells recognize them. Mm-hmm. And then make more of that peptide to give it as a vaccine against the individual tumor. So it's a personalized vaccine approach, and it has seen some success. I think Steve Rosenberg was the first to do it with melanoma. It hasn't been universal. We haven't worked out all the rules on it yet. But yes, cancer is it's based on changes in genetics.
Another question regarding genetics and modification comes from user XN4W. I'm not going to read it. Huh. Although that was my choice for my second child's name, user X. Okay, he or she's asking, do GMOs, genetically modified organisms, affect our DNA? If so, to what extent?
Only through the plasticity of your brain be infected by that notion that they do anything negative.
Okay, this is a possibly contentious one. This comes from a man whose name is an aptronym, Gene, like your friend who came up with the algorithm that you mentioned, a suitable name for his job or her job. He or she is asking, why weren't you able to create life? What's the missing spark? The reason this is a good question is that I don't know if he'd argue that he did or didn't. So did you create life? If so, what was the missing spark? Or if you did not, what is the necessary lacunae that is missing?
So right now, all life that we know comes from pre-existing life. And even the synthetic cell we made a totally new species that never existed before, but we used an existing cell to transplant the genome into, and that genome converted that cell into totally a new species.
Mm-hmm. So it's prokaryotic. It was a cell wall. You used the existing.
Yeah, it was a bacterial.
Yeah. Mm-hmm.
And mycoplasma, so there was no cell wall. On the same day of our announcement, I got comments from President Obama and from the Pope.
This is in 2010.
President Obama announced that he was forming his new bioethics commission to review the ethics around making synthetic life. The Pope said, Dr. Venter did not create life, he just changed one of life's motors. So there are groups with the notion of trying to make it from a cell-free system. We're trying to do that. But in my book, people talk about making life from scratch. And I use the analogy of people making a cake. And when they talk about making cake from scratch, instead of buying a cake mix, they buy a bag of flour and they put the different things together. It's not like they went out and planted the seeds, made the flour, made every component to it.
Well, as Carl Sagan says, to make an apple pie, first you must create the universe.
Yes, yeah, that's true. And so I think making it from cell-free components will happen in the next few years. We're hoping even sooner with experiments that we're doing at the Venter Institute right now with the synthetic chromosome and trying to boot that up. But it's still not doing it from scratch. Instead of an intact cell, we're taking the components from a cell and trying to do it. But it would at least prove that you can form a new cell from scratch by having components of lipids and everything there. There's been groups for decades trying to put proteins in lipid bags to see if they could get a living cell. I've told them it's impossible. They can't possibly do that because you can't have life without DNA. Because what we talked about earlier, the proteins turn over in seconds to minutes. You can't have a living self-replicating cell without the ability to remake all the components on a continual basis. So I would say we definitely created a new life form that has never happened before by humans in history. And we've shown the principle that the genetic code is the driving force of life, and that you change the code, you change the species. We're trying to do it without a cake mix, but it still gets down to definitions of, well, you didn't make the ribosome yourself, you didn't make these other proteins yourself.
Right.
So
you didn't make the DNA.
The fact that you can convert life by writing the genetic code from scratch, we start with 4 bottles of chemicals, I think makes it more of a theoretical argument than an actual one, but proving that we can take just the genetic code with components and get a cell de novo, I think would be a nice addition.
Yeah.
Genetic technologies, including gene therapy and synthetic biology, are surrounded by ethical, social, and safety concerns. However, despite being controversial, they offer promising opportunities to revolutionize medicine, improve personalized treatment, and tackle various challenges of human life. So it's essential to stay up to date with the latest developments in this exciting field. For instance, recently the FDA approved the very first treatment to use the gene editing tool known as CRISPR, which my wonderful guest on today's episode, Craig Venter, talks about. They just approved it. But the media landscape surrounding these developments
is often fragmented, making it hard to find reliable information and find a balanced view, which is personally why I love ground news so much. I even deleted the iPhone news app that's default on my phone and replaced it with Ground News' widget app, and I also bought it for a present for my older brother Kevin. Ground News is an app and a website that gathers together all the world's media into one place so readers can compare coverage and see the full picture of what's being reported worldwide. For every story, you get a quick visual breakdown of the news outlets reporting it, including their political bias, how factual the source is, which entity owns the source, and which countries are actually reporting and covering the story. For example, let's return to the FDA's approval for the first CRISPR treatment. Right away, you can see that 162 news outlets have reported on the story. Of these 162 outlets, 23% lean left, 11% lean right, and an overwhelming 66% come from the center. You can even see who owns the outlets reporting on the story.
63% are media conglomerates.
Ground News also makes it easy to compare the headlines to see how the bias might shape and influence the framing of the story and affect our understanding of it. One of my favorite features of Ground News is the blind spot feed, which allows you to see stories that are underreported by either side of the political spectrum. Ground News lets you get a deeper understanding of the complexity and nuance of different issues by identifying media narratives and their biases. You can see every side of the story and thus develop a well-rounded worldview, which is pretty substantial in these turbulent times. You get access to newspapers and things that are very hard to find. Go to ground.news/Dr. Brian to stay fully informed on breaking news and compare media coverage. Sign up or subscribe through my link for 30% off unlimited access if you support the mission, and find it as essential as I do to scientific literacy.
Okay, there's 4 questions from this person named Cruz. First one, how resilient are cells? And now he's bringing up Michael Levin again, a different person. Michael Levin says they are quite remarkable in adapting to harsh environments. Are cells intrinsically resilient?
There's no universal statement about it. We have microbial forms that live in pH 11 water. We have ones that live in extreme acid pH 1 or less. If you put your finger in the water, the skin would dissolve. So these organisms live in that. We have organisms, I said earlier, that live up to 135 degrees centigrade. You know, we would die very rapidly at those temperatures. Proteins sort of fall apart at 100 degrees centigrade, but in boiling water. And so we're beginning to think it's the boiling action that mechanical action, not just temperature alone, that causes them to fall apart. And when we sequenced the first archaea, it was a methanogen with Carl Woese. This is methanogen, grows about 85 degrees centigrade, lives by converting CO2 into methane. We have the extreme radioactive radiation-resistant organisms, Deinococcus radiodurans, that can take huge doses of radiation. Basically, the radiation does break the chromosome apart, but it seems to have extraordinary DNA repair mechanisms for putting it back together. So the answer is yes, we can find life in the most extreme environments that you can imagine that don't work for a human-centric view of life.
So this one's kind of a yes or no question. Could we terraform Mars? And if so, what life form would do that? And I would add a question, could we re-terraform Earth, in the sense, could we develop a life form that could scavenge, as well as a tree does, the CO2 in the atmosphere and reverse, carbon capture. Anyway, how would you, if you had Francis Collins' budget and Anthony Fauci's budget, how would you use that money?
Francis Collins doesn't have a budget anymore unless his wife gives him one. But
how would you terraform Mars and Earth?
Well, keep in mind that Earth didn't start out with an oxygen atmosphere. Our oxygen atmosphere came from life. So whether it came from panspermia life or just spontaneous versions here, eventually the cells that convert light into oxygen and CO2 are why we're alive today, and big portion of the organisms that we've discovered in the ocean. In fact, we found all kinds of other ones that deal with light and energy. If we wipe those out, we wipe out our ability to live here.
Mars doesn't have an ocean. And
It supposedly has subsurface oceans that still has to be totally proven, but I think the data looks pretty good on that. And if there's oceans that have seawater in them and they have oxygen or even no oxygen, we will find life forms there.
Do you think it's possible to terraform Mars? I mean, with biology alone, or
So, as we discussed a little bit earlier with people and going up there and things we're sending up there, we're already contaminating it. Whether we terraform it into, an Earth-like planet or, a cesspool remains to be seen. So it depends what you mean by terraform.
I guess make Earth 2.0 like Elon's, hoping.
If, one of the things that the planetary scientists are discovering, they're discovering all these Earth-like planets. Some are just straight oceans, 100% ocean.
Yeah, water worlds.
And they certainly have life. If not, we could send, a few inoculations up there and get some photosynthetic organisms going there. So yes, under the right circumstances, I think it's totally possible to do that.
I want to run an argument by you. I've used it on folks from Joe Rogan to other individuals. And that's the following. If I tell you my colleague upstairs or down the hall from here in the astronomy department at UCSD, I tell you that she's discovered a double planetary system, at least two planets. They're both rocky core. We can tell from their mass, their density, their orbital periods, and their distance from their host star, which is a type 2 G2 subdwarf, just like our sun. They're in the habitable zone of that host star. If I tell you there's two planets there, and one of them we see, there's like blinking lights, and there's like, looks like, superconducting, levitating monorails. And then I just asked you, just knowing that there's one of these two planets is teeming with life, technology, all different speciation and so forth. And I said, Craig, can you tell me, what would you guess the probability is that the neighboring planet, which we just haven't had time to point the Webb telescope to, but she'll get to it. What would you guess the probability that that planet would have life, also in the habitable zone of that same host star system?
So let me rephrase it a little bit. I'm certain that we will find microbial life ubiquitously throughout the universe. The arguments that I've heard that make sense to me, that expecting evolution to create higher life forms, assuming that's what we humans are, there's a lot of random things that happen that probably helped higher life evolve here, such as the mass extinctions, right? And the various events that took place. So it's, there's not a simple recipe that this was a linear track that we went to from a viral microbial world to higher life forms.
Right.
But just like my idea that life happens, multicellular life clearly does happen. If you look at the sheer diversity of what we have and these things that are found in the deepest high temperature vents now, some of these animals are just incredible looking, like they, better than anybody's imagination for what space creatures would look like. So it's really a question of rates of evolution and conditions for that. If it hasn't happened, it will happen. And it's just, it's more in your realm. Has there been enough time, is the universe too young to have that happen multiple times?
I guess my argument that I keep trying to make, and people push back on me, is I say we must be able to, in a Bayesian, prior updating your prior sense, it must be possible to say something about the fecundity of life in the universe and its resiliency based on the Earth-Mars system, and that we know there are these two. So the system I was talking about is obviously the Earth-Mars system, and the fact that we haven't explored every square millimeter of Mars, but of course, and you brought this to my attention just now, the underwater, potential evidence for underwater ocean, the extremophile, vicinity of extremophiles on Earth, deep smokers, all the stuff that you've mentioned today, acidic life, radiation life, carbon dioxide, philic life, anaerobic bacteria. And so the fact that we haven't seen, it's impossible to say that there's absolutely no way we can use the non-observation of life in our solar system outside of Earth. And I think it's controversial. I don't understand why it is. I mean, I have colleagues like Lee Cronin, who's been a guest on the podcast remotely. I've never met him, but he thinks there's life everywhere. And they kind of use this Carl Sagan argument from Contact with Andrewian, who I had on the podcast. She's an amazing person. And that's, well, if there is no life, it's an awful waste of space. And I say, well, I went to Antarctica twice, and I saw almost no life besides the red, patch-wearing penguin-like creatures, including scientists. And that was it.
But if our visual acuity was different,
yes,
and you could see what I saw,
you're right,
with DNA sequencing.
Yeah. I guess I mean technological life.
The planet would, if we could see microbes. I mean, because I've gone diving and swimming in some of the most pristine, crystal clear water that has 10 million viruses per milliliter in it, right? It's beyond our visual acuity.
Sure.
And so we don't have the senses to detect life on our own planet. We've had to develop specialized tools to do that. And those have only happened in my lifetime. I don't think we've done the right experiments yet to look for life on Mars. They're trying to. They're going to be having a drill that drills down a little bit.
Yeah.
It probably won't get down to the subsurface oceans yet. That it just need bigger equipment and things to do that. We haven't sent a sequencer up there yet to try and look at things.
I guess, you're 100% right. I guess my thought is...
But there are high energy bonds. There are, chemical signatures of life found almost every place that's looked.
Oh yeah, no, I stipulate there's life in the universe. I do think it came from the Earth, I think we're in violent agreement about that.
Or
I guess
it came from Mars and Earth was terraformed.
And then it terraformed, right?
Yeah.
But the question is, are there advanced technological life form? I think that's the interesting question. And some have proposed many different ways that could happen, and not see them. And there could be so many different variables. But I guess again, it has to be the current understanding has to be that we don't know of any. And I think we have to be guided by it. We have to be open-minded, and people in my channel will be screaming, like, why are you so closed-minded to UAPs being actual extraterrestrial intelligence? And I say, look, no one would be more excited for there to be actual aliens than a physicist, right? And probably, molecular biologist, kind of a physicist.
I'd love to do their genome first.
Yeah, you'd love to, right? I'd love to know the physics of the 28th century, But, today, when you look at what are the actual, kind of proof texts given, is there some barrier? I always joke, you'll make fun of me, I'm sure, but we couldn't be having this conversation now. I mean, it might have happened at some point in human evolution, or in the evolution of technology on this planet, whether by humans or some other creature. But we are using these lights, and these are kind of rendered impossible, but for the fact that we had a lot of whales that lived on the planet. And we had a lot of, these primordial bacteria that made, so if we didn't have that, we wouldn't be, it's not like you could make a solar panel from a solar panel. Like the first computer wasn't made on a computer, it wasn't designed. So to what extent are these things, dependent on or contingent upon, the exact circumstances that we had with, early explosion of life, and then the petrochemicals that enabled the industrial evolution, which then enabled the information revolution.
So the thing think about a couple of things, just probabilities and the rate of genetic mutation. So one of the biggest findings that we talked about in the book was that in the ocean, where it's exposed to UV light and oxygen, instead of single punctate organisms that we're used to of classification, we found these huge clouds of tens of thousands of closely related species, all doing photosynthesis, all generating oxygen, but differing as much as 50% in their genetic code.
Wow.
Now, we're plus or minus 5% from all other mammals. Right?
Like orangutans were 1%.
So if you think of that diversity, gene order and function is maintained. And one of the biggest discoveries we made was that the viruses, so the phage that sort of regulate the microbial density in the oceans, they don't want to kill their host. Mm-hmm. In fact, the most profound discovery was that the phage take in new copies of photosynthetic genes to upgrade the software of their host to keep them really robust. Mm. So it makes you think philosophically, well, then are viruses in charge? But viruses are not living entities
teleological
and can only reproduce in their host, but they keep their host alive to do this. But the mutation rate is really high. So our synthetic minimal cell, a group took it through 300 cell divisions and measured in that time. So that's equivalent, I think they said 20,000 human lifespans. Mm-hmm. And the cell underwent a very high rate of mutation and became a much more robust organism just in this short period of time. From the minimal cell that we had created, it became a much more robust life form. Mm-hmm. So when you understand these rates of mutation, particularly caused by UV light, by radiation, by chemicals, I think it's virtually impossible not to find higher life, at least multicellular life. Mm-hmm. And once you start multicellular life, you're on the route to higher life forms, because multicellular life needs to have coordinated control centers. That's how nervous systems develop. It's like the oceans, what people thought of them 10 years ago versus what our studies showed. Somebody asked me, what ended up being a kind of profound question. So you proved all these things that we didn't know exist. What else don't we know? Yeah. and so
the unknown unknowns.
So we don't know what we don't know, but it has to be a lot. Just like we couldn't detect these before DNA sequencing, we don't necessarily have the means to detect something 1,000 light years away, whether there's intelligent life there or not.
Mm-hmm. Is there a problem with information overload that you're gonna just have? Like you said, we only know 1% of what the human genome, and that's been sequenced for 20 years thanks to you. So collecting more and more, is it just kind of like, I can't even think of the analogies, like one of my kids just collects Pokemon cards and he'll never be able to do anything with them because he's going to run out of interest before he runs out of available Pokemons, right? So is this information overload?
Well, it can be until there's new tools developed, right? I mean, and so in your field, you're looking for new methods to pick up new signals. Yeah. I think the most profound discovery in history would be to not find life everywhere else. I think that would be the more disturbing finding than to find it ubiquitously throughout the universe. And just statistically, now that we know how many Earth-like planets there are in the Goldilocks zone, I'm certain there has to be higher life than just microbial life.
Mm-hmm. What would count? So I've asked this in the context of my cosmology friends who are now, along with myself and my team mates on the Simons Observatory, we're poised. There are 17 fundamental elementary particles: neutrinos, quarks, electron, photon, Higgs boson, etc. 3 of them, we don't know their energy or their mass energy levels. We know that there's a bound. We have a lower limit and we have an upper limit, but we haven't detected their mass. They're the neutrinos. We know they have mass from neutrino oscillation experiments. We know from cosmology that, and from laboratory experiments, that they have an upper limit, that they can't be bigger than, or else they would have been detected and measured. So I asked my high energy collider physics friends, the guys, the Lawrences, the Fermis, etc. I say to them, what degree of belief will you have when we on the Simons Observatory, hopefully in 4 years' time or less, we tell you the mass of the neutrino is exactly 0.0275 electron volts, 27 electron volts, which is consistent with upper and lower limits. Yes. Will you believe it? Because now we're on your turf. Now we are infringing upon the land last trodden or first trodden by the Lawrences and the Fermis and the Oppenheimers, laboratory collision physics. We're now using the cosmos as our particle collider. Will you trust us? And I get about 50-50. Half of them say, yeah, we'll believe it. Half of them say, no, unless I can reproduce it in a lab experiment, I won't believe you. I want to ask you, my astronomer friends, see a signature of, methane or, some kind of weird, carbon dioxide or something like that on another planet.
Carbon bonds.
Yeah. They see a benzene ring or whatever.
Yeah.
And it's not like Aldrich or, will you believe it? Will you believe that? What will it take for the whole biology community to accept that an astronomer has made a discovery of their domain's expertise, i.e. life? DNA? What would it be?
But that's happened over and over again, in my view, that these chemicals are found. And, my view, every place we have the same chemical components, we're going to have life.
But we won't be able to do a lot. I mean, we made chemical experiments. We can go to the salt flats of Chile, and then we can go to Old Faithful, and we can say, oh yeah, they are extreme. But we can't say, well, here's this chemical. It might not be DNA. It might be something else, right?
It's most likely going to be DNA because we find the nucleic acid building blocks out there. We find the protein building blocks out there. We need tools that maybe could detect polymers of those things but the bond does show you that there's connections. I assume those things are widely accepted. I didn't know anybody doubted it, but I guess in this date of people not believing science at all.
I'll just say, like, if you couldn't throw it into shotgun sequencing, or you never be able to touch it. It's 80 light years away.
Well, science is a very bizarre field, particularly compared to the political domain today. And there's a lot that's flawed in science. We're up to 25% of papers are fraudulent to some extent. And with ChatGPT, I'm sure that's going to get worse. There's 20 times more scientists alive today than when I got my PhD at UCSD in 1975, which means it's 20 times harder to get a Nobel Prize, presumably, or other things. But
infinitely, in my case. Yeah.
But the ethics of science, at least what I was taught here by the best biochemists in the world, was an essential part of science. And that's slipped away, and it's totally slipped away to nothing in politics and the fake news. And, people will believe their soap opera, but they won't believe what comes out of science. There's a lot that's wrong in science. And I was citing things earlier like junk DNA and other notions like that. The difference between science and politics is science is self-correcting.
Yes.
You can have these crazy bullshit ideas that you wonder how they ever develop. And science is not infallible because it's done by people that, want their story, it's part of their fame to become dogma. But it's self-correcting. And, the Voyage of Sorcerer corrected more dogma in one set of experiments than a lot of things have. And to me, that's the real pure beauty about science. It is self-correcting. It's loaded with false ideas and false notions and limited thinking because we didn't have the data. That's why people thought there were 300,000 human genes. As soon as we had the data, that changed overnight. All these things will eventually be self-correcting. How much of society believes them is a different thing than at least the upper end of the scientific community, the people that are doing science and putting ethics first, which I hope is still the majority of scientists, but it's, we're being challenged all the time with these notions. So the self-correcting nature of it means that all the future discoveries are going to be mind-bending.
Yeah. So one last question from the audience. This is from a friend, Greg. He was asking about your thoughts on this new discovery or this new gene editing treatment that cuts dangerous cholesterol. It was a very small study, but I guess that scientist, this is at Verve. Therapeutics in Boston. They announced that a treatment appeared to reduce cholesterol levels markedly in patients. It appears to be safe, but there's only 10 patients. I guess that's better than 6 rats or something. But what are your thoughts about this? Would you ever undergo gene editing for yourself?
So there's two kinds of gene editing. Gene editing that's done ex vivo. So you take cells out of the body, you change genes. So this is being done for new approaches to cancer chemotherapy, a lot of diseases. It's very effective. It's, in my mind, totally ethical and reasonable to do. The other type of gene editing is germline editing, where you change your genetic code for all future descendants of you.
In the sperm or egg.
Yes. And that's what we're not ready for. We don't have enough knowledge to know how to do that. People are still with limited thinking that there's one gene, one function. In fact, there's pleiotropism is the rule, not the exception. We did editing of the pig genome and to get pig organs for human transplantation.
Can you get them to chew their cud? Because I would really like to start eating them, and I'm kosher, so I can't. But if you could edit their genes and make them chew their cud.
Well, so we put human genes in the pigs. So I don't know if you eat bacon from those. Is that cannibalism now? But we've changed it so that the organs aren't immediately rejected. There's, we've only changed 10 genes so far, but the 2 heart transplants that have been done with our 10 gene pig hearts lasted longer than the first human to human transplants. Wow. But they still didn't last for a super long time. There's 1 million people that need kidneys a year in the US and can't get them. And so we're trying this approach, but the reason I'm starting this, so I sequenced the strain of pigs that we were using, and we looked for a place, what we call a landing pad, a place in the genome that's not subject to recombination and genetic variation. So we could insert new genes there and not have them be part of rapid evolution and changes. We don't know enough yet about how the human genome functions. We're lucky these studies with the pig worked out, and I think it's because we did that preliminary work. CRISPR is a wonderful discovery and a wonderful tool, but a lot of people don't understand the fundamentals of it. CRISPR has what's called off-target effects. So it's used on, you use DNA primers. These are just short pieces of DNA sequence. You pick them that recognize the site that you want to change. But in 6 billion base pairs in the human genome, there's multiple sites. So they're called off-target effects. So people look at the gene they're interested in, they see they're making the changes they want. But when we use CRISPR editing the pig, we resequence the whole genome to look for off-target effects that could be knocking out another gene. So we're not ready to use CRISPR alone to do germline gene therapy. So all these things sound great. People have talked for decades. When I first started in the human genome, that's all I ever got on TV interviews. Are we going to edit the human genome? And genome ethicists like to talk about, the ethics of doing enhancement versus getting rid of diseases. So it's a space we're very early on and not ready for germline changes.
Got it. Well, we've reached the point where my bladder will not allow us to continue beyond these final 3 questions. And they're all in some way related to Sir Arthur C. Clarke. And you had that conversation with our director, Eric Veary, recently. And so you know that Arthur C. Clarke was very quotable. And in fact, we'll get to the origin and the name of this podcast at the very end of these 3 questions, the final one of the 3. But the first one harkens to his famous statement that any sufficiently advanced technology is indistinguishable from magic. And that's a quote we open every podcast with because we have him recorded in his own voice. Yeah. And I want to ask you, of all the inventions and all the discoveries that human beings have made, either yours or others, which one do you find the most magical and the most bemusing and delightful of all human technology.
We were totally stunned when we transplanted the chromosome and it booted up and created a new species. That
It's pretty cool.
It's what we were hoping for, but it was pretty cool
To actually see it happen.
I depend on breakthroughs in physics and chemistry with trying to characterize the phenotype of individuals. I'm blown away by how effective MRI works by looking at vibration of water molecules and how that can diagnose cancers with some of the post-processing algorithms that were developed here at UCSD. So, when it's not part of the repertoire we can do with our hands or we can see with our visual acuity,
that's right.
It of course all seems like magic because if you're not using those tools, you can't see it.
That's right. That's what my favorite quote from Galileo. He said, you should measure what's measurable and make measurable what's not yet. So I think that's exactly in line with what you said. Okay.
Oh, that's Galileo.
That's Galileo. Yeah, that's Galileo, Einstein. I've got Carl Sagan. I only have one of a person who's actually on the podcast, and that's Noam Chomsky. It's somewhere over there. I can't find it.
I should have brought my bobblehead that I was.
Oh, yeah. I'll expect that for you around too. Or I'll come visit you in the JCVI. Okay, next question is another quote from Sir Arthur, and he said the following. He said, when an elderly but distinguished scientist says something is possible, he is very likely to be correct. But when he says something is impossible, he is most likely wrong. And I want to ask you, not calling you elderly or what have you, but I want to ask you, what have you been wrong about? What have you changed your mind about? What do you regret, if anything?
Well, I'm on my third marriage.
well, the second marriage is a triumph of hope over experience. What's the third marriage?
So I learned from my mistakes and
iterated.
I'm a
scientific method.
Slow learner, and I finally got it right, but it took a while.
Yeah.
I think what I've been most wrong about, sadly, is gauging people and their motivations. The most delightful experience I had was at Solera, where people just came in, wanted to be part of it. They believed in what I was doing. They wanted to be part of history. They wanted to contribute their knowledge to that in the purest fashion. And everybody exceeded even their own expectations of themselves with doing things. That was the best of scientific humanity that I've ever personally experienced. When I started human longevity, I naively kind of expected the same thing. But when I started Solera, the world hadn't heard of me. Other scientists had, but it was pretty much an unknown. And now I was a known name, one of the 10 most known names currently in science. Yeah. And despite what people said, their motivations were clearly very different to come and join the project to try and mix phenotype with genotype and create human longevity. And I take people at face value, which I learned is not always the wisest thing to do, but and my mistake is reading my motivations and ideas into other people. And I've been helped. You've read my book, so I was thrown in the brig twice. I've had some wonderful second chances and second opportunities, and I constantly believe in giving people second tries and second chances. It works out probably 1% to 2% of the time, and that's a disappointment, because it was so profound in my own life. Yeah. So I'd say my biggest failing it's been my biggest success, and my biggest failing is selection of people to work with.
Mm-hmm. So the last question actually maybe touches upon what you were just saying, but maybe we can elaborate a bit more, and that's the final of Sir Arthur's famous quotes, which is actually the origin of the name of this podcast. And he said, the only way to know the limits of the possible is to transcend them and go into the impossible. That's my podcast name. I want to ask you, in the form of advice to your younger self. You speak candidly about your time in Vietnam and how you actually attempted suicide by swimming out to sea, and obviously it didn't work, thank God, because the world would have been devastated, right? I want to ask you, if you had 30 seconds with your 20-year-old self, what would you say? What would you give him to give him the courage, rather, to go into the impossible?
So I actually learned the lessons that are key to my success probably in Vietnam. I learned some of them earlier being a competitive swimmer. I was a lousy student, and I know now why. It's because I have aphantasia instead of a photographic memory, and the school system selects for photographic memories. And so I nearly flunked out of school, and part of believing in myself is when I started from nothing and became a champion swimmer. And that, sports do give people a lot of self-confidence because you learn physically what you're capable of. And I would beat people that were technically much better swimmers than me just because I was more motivated to win. My mentor here at UCSD, Nate Kaplan, said the biggest failing that most scientists make is they talk themselves out of doing the experiment. And that's true. You see it over and over again everywhere you look. And some people are afraid to succeed. Some people are afraid to fail.
The imposter syndrome.
It covers all the range of things. In Vietnam, I learned all I had to lose was my life, and came close to doing that a number of occasions. One, as you say, was potentially self-induced, but a shark told me to wise up. I came back probably more fearless, than was good for me. Reporter from Nature who read the book and was writing a review on it, she goes, you had a lot of near-death experiences in this voyage. And I said, well, they weren't planned to be that. I've learned to believe in my unique type of thinking, the combination of conceptual thinking with intuition. I've learned to trust it. More importantly, I believe in myself and my ideas. And it's a narrow range because you slip a little bit one way, they can become delusions of grandeur, and you go off the crazy spectrum. I think I'm successful from having tremendous capabilities of doing things myself. One of my biggest problems in science was giving up doing the experiments because I could do them so much better than anybody else and taking over on things. But it's believing in yourself, and, failure is nothing compared to losing your life, for a worthless cause.
Yeah. And seeing it through those lenses, and it's quite emotional to hear you speak with such candor about it. And to me, I'm thankful that you saw the light, or that shark intervened, and however it happened serendipitously, we'll never know. But they say, I believe it was Schopenhauer, or no, it was Kierkegaard. Kierkegaard said life can only be understood in reverse, but unfortunately it must be lived going forward. Yeah. I want to thank you, Craig, for your so much of your time and your influence on me and millions of people. And call you the Merlin of the biological realm. So thank you so much for sharing your time with us.
Thank you for having me.